So first, thanks for the opportunity to introduce my work on computational study of gene regulation in the era of next generation sequencing. So my name is Haiyan Hu, and I'm currently an associate professor in the Department of Computer Science. So, I majorly work on computational methods to understand the biological and biomedical data. So my lab is called the Data Integration and the Knowledge Discovery Lab. So, this name largely reflects our research methods and the goal. So I came to UCF in 2008. In the past 12 years, I taught both graduate and undergraduate courses. So the graduate courses I taught like Introduction to Bioinformatics and Machine Learning for Biomedical Data. So the undergraduate courses I've taught include courses like Introduction to Discrete Structures and the Computer Science 2 core course. So, let's talk about what motivates the research. So it most comes from the Human Genome Project around the early 2000s. Okay, so Human Genome Project is quite famous, right? So the goal for the Human Genome Project is quoted as imagine a world in which we will be able to treat diseases by altering our own our very genes, giving us new ones if ours are non-functional, changing bad genes for good ones. For the first time in our existence, we are closer to understanding just what we are, and we now have the tools to make the whole world better through science, the science of human genome. So this message is very powerful, and it brings about many complicated questions. One of them is how are genes regulated in a cell? So we know that genes are transcribed into messenger RNAs, which is then translated into proteins. So if we want to change our genes, so we need to understand how the genes are regulated under different cellular conditions. So there are a lot of ways to control or regulate our genes. For example, the transcriptional and translational gene regulation, so which occur during the gene transcription and the translation procedure, respectively. So, because the NGS, next generation sequencing, so recent years, we have seen a lot of data, genomic data, omics data, different types of data. So by 2025, so over 100 million of human genomes could be sequenced by 2025. So with so much genome-wide data available, so computational approach has become an indispensable part. Let's see this. Okay. So the good thing is that there are patterns upstream of the genes in the DNA, so that are associated with gene regulatory mechanism. So these patterns are often called DNA regulatory motifs. Computational people like patterns. They are good at patterns. So one of our focus is to develop a computational approach to systematically identify DNA regulatory motifs from NGS data. So we have published more than a dozen journal papers on this topic. A unique aspect of our methods is that we try to identify the frequent co-occurrence of multiple transcription factor binding sites in short genomic regions. So different from existing methods, our approach can consider any number of binding factors simultaneously and can predict a large number of genomic sequences, such as from the ChIP-seq data. So for example, one of our tools is called CellMix. So CellMix can predict motifs and binding sites of more cofactors in more accurate and time-efficient ways, comparing with currently existing methods. So it has been used to systematically discover motifs and binding sites of transcription factors and their cofactors from ChIP-seq data. So we also used the CellMix and performed a comprehensive motif analysis on the prognostic gene signatures from 5 cancer types. So we demonstrated that gene signatures from individual cancer types, as well as across different cancer types, share regulatory motifs. So we also observed that transcription factors are likely to bind these shared motifs, also have prognostic functions in cancers. So because people need this computational resource, computational information, so we have generated a complete, nearly complete list of human DNA motifs under different conditions. We used 349 human cell and tissue samples, use their DNA-seq data. So another focus of our research is to understand the non-coding RNA gene regulation. So one of the major non-coding RNA is microRNA. So microRNA is a very small RNA, and it has powerful effect on gene regulation. So microRNA regulate their target genes through binding interactions. So we have integrated the most recent haptoput data to study features of the binding interactions. So a lot of algorithms have developed in the recent years for this same purpose, but there's a lot of false positives. So we wondering, so what will be the missing features that lead to those false positives? So we have created a set of computational methods and tools try to analyze the recent class data or CLIP6 data and try to identify what caused those false positives. So we have, based on which we have identified a feature called, we call it DPS, which use the Markov chain property of the position-wise bonds and relationships between adjacent positions from experimental data. So we also use the different machine learning approach, and we developed a computational approach try to consider the relationship between microRNAs, like they're interacting in a different way. For example, they compete with each other. Sometimes they cooperate with each other anyway, so they're not working individually. So those work have led to a set of softwares and tools which can help reduce the false positives in current approach, and it's also offer those tools for biologists to use for microRNA target set prediction. So, so another project we are recently interested in is gene transcription initiation and its impact on gene regulation. So we have performed a comparative study to unify microRNA TSS or transcription start sets with proper interpretations. So those studies have generated or produced a database that integrates the microRNA TSS set in 14 recent large-scale studies. So we also developed deep learning-based methods such as DLCage and Dept. So those are our names to model transcription start set and predict microRNA TSS. So those methods obviously result in tools and the database for people interested in transcription initiation. So that is pretty much, but it's not a very detailed explanation, right? So if you're interested, you may consider read our papers, and we can propose new projects. So talking about the goal in the recent years, we would like to focus on, continue focus on NGS data integration and analysis, and try to understand the gene regulation under specific conditions in specific diseases. So in the long term, so we try to understand the biological system at multiple scales, the cellular, tissue, organ, and system. So we want to further develop data analytical methods for personalized medicine and healthcare. So finally, so the above work is majorly supported by multiple awards from NSF, and we also want to acknowledge the members in our data integration and knowledge discovery lab. That's pretty much of it. Thank you for your attention.